And the reason for that is because of the markets. So the invention and discovery that came from the measurement of calcium and then the replacement of radioactivity had 2 or 3 impacts. First of all, we discovered a mechanism which is very important in rheumatoid arthritis. Calcium is the switch inside the cells that releases toxic substances that cause a joint to be damaged. And also we proposed that it was the mechanism that causes the myelin in the nerve, which wraps around nerves in the brain, to deplete in multiple sclerosis. And you can see these papers developed with colleagues in Cardiff were published in the world's premier journal, which is Nature. But the other method had even more impact. And really simplistically, it involved a bit of chemistry, where this compound was developed as a model by a colleague of mine in Sussex to model the jellyfish flash. It works by reacting with peroxide. The problem was to get it to flash. Because there's a competition with these 2 moieties here, I suggested doing the reaction in acid to start with. And that turned out to be worth about £20 million of income into Wales. Because this technology is now used in several hundred million clinical tests per year. It has brought in royalties and a very large amount of commercial income developed by colleagues of mine who set up a spin out in Cardiff. Won a Queen's Anniversary Prize, was highlighted in this project as one of the top inventions and discoveries in the last 50 years from British universities. And last year there was a Russell Group report on projects from Wales that have had economic benefit. The only one they selected, from the UK, sorry, the only one they selected in Wales was the bioluminescence one. But the important thing is it's transformed clinical diagnosis. It isn't just about money. Because there were loads of labs around the world that couldn't do this method because they couldn't use radioactivity. The best test for AIDS uses this method. So I got curious about colour, as I said earlier, and I discovered there are 3 ways that nature produces colour. We have a complete rainbow, from blue right through to red. Uses different luciferins. So fireflies emit yellow, jellyfish emit blue light. There are some animals which produce a different solvent around the excited state. You can demonstrate this with fluorescence. So if you put this molecule into a solvent, not like water, hydrophobic, under UV light it emits blue light, but in water it emits yellow light because the energy levels are different. That's why a firefly emits yellow and a glowworm emits green, even though they use the same chemical reaction. And then there are flaws associated with bioluminescence, because it could be the luciferin, it can be the product, which is the emitter, but it can also be the other way that animals produce light by different colours, as I showed you earlier, a fluorescent molecule. This molecule in the jellyfish is called the green fluorescent protein. I'm sure you teach about it. And again, quite amazingly, it's led to $1 billion market. And it turns out that these jellyfish, a lot of them have these green fluorescent proteins which have slightly different structures. And this is what it does. What it does is it changes, this is the spectrum, where it's blue, it shifts it to being slightly green and sharpens it. And here is the green fluorescent protein in the jellyfish. So it seemed very, very obscure until what was discovered was that if you genetically engineer it, then I started at 1:15. It gave me 50 minutes. I'm going to have my 50 minutes, sorry. So 2 years ago, it led to a Nobel Prize just because somebody got interested, curious about how a jellyfish produces different colour. I will speed up for you the story. And unfortunately, I missed out on this, but there we are. It produces different colours by genetic engineering. It also teaches us why corals are coloured, because they produce similar proteins which make the colour molecule from the protein sequence. But the most important reason it's led to billion-dollar markets is because when you express the DNA in a cell, the cell becomes green fluorescent, and you can use it as a marker. So I looked at 3 different ways to produce different colours. I call them rainbow proteins, and this set enabled me to set up a spin out called AK Rainbow to develop the potential of technology. One of the potentials is a traffic light indicator for environmental toxins, including agents of terrorism. There is no proper detector for frontline responders in the event of a terrorist attack. This technology will do it. Unfortunately, there's some idiot in the Ministry of Defence that won't listen to me. This is the way you can detect these in seconds, and it will give you a traffic light indicator, but I cannot get funding for it. So I call them canary proteins with the analogy of canary in a mine. It also is useful for drug discovery, because if you're looking for a drug to kill a cancer cell, the drug that works will convert the cell from emitting green light to emitting blue light. So it gives you a traffic light indicator of the drug you want to develop. The method enables us to measure things in live cells. So these are pseudo-colour. So this is the Firefly expression measuring ATP in live cells. It teaches us why the trees and bushes in Wales are bent by the wind, because the wind generates calcium signals. This is lit up as a result of putting the DNA from a luminous jellyfish into an intact plant. So this is the cliff top where I live. So the hypothesis is that the wind generates these calcium signals which make the cells on this side of the tree grow slower than that. As a result, the tree protects itself against the prevailing wind. Now, of course, this hasn't been done in an oak tree. It's been done in a model system, but that's the hypothesis. It also gives us a way of looking at gene expression. So this is the best way of looking for drugs that will be used to treat cancer. So a huge billion-dollar market again, used by the drug companies. They link the gene they want to switch off with the drug to the DNA that makes the protein in the firefly. And you look at individual cells, or you can look at a whole animal like a mouse. When the drug works, the light goes out. If the drug stops working, the light comes back. And the other thing which is remarkable is you can actually measure things in different parts of the cell. By using genetic engineering, we targeted these proteins to the membrane of the cell, the cytosol, to the nucleus, and to the endoplasmic reticulum. And this is now the green fluorescent protein. And this also destroyed a myth that I was taught as a biochemist in Cambridge, that the endoplasmic reticulum, this structure which wraps itself around the nucleus, is vesicular. It's not. It's a three-dimensional spider's web, and you can see that it's moving. So using the green fluorescent protein, we can light up structures in individual cells. So finally, in terms of the science, one of the things I've been working on for 10 years is the illness that afflicted Darwin. Not many people realise he was ill. He was ill for 50 years. It's why he moved out of London. And we believe he was intolerant to milk. This is going to have a major impact on clinical management of the commonest illness, GBC, which is irritable bowel syndrome, and again will inflict on billion-dollar markets.